my work focuses on aging. I look at cognitive and structural brain changes that happen as we age, both in normal aging as well as pathological changes like in Alzheimer's disease. And I do extend beyond the traditional boundaries of just looking at cognitive by looking at health and social factors and how they all interplay together to influence our cognition and brain structure. So as you can see, based on the title today, I'm going to talk about divergent paths in subjective cognitive decline. So essentially, how do different methods that we use to classify or define subjective cognitive decline result in different brain patterns? So is there differences based on the questionnaires used? But to talk about subjective cognitive decline, first we need to do a little trip down the aging journey. So with age, there is this normal trajectory of cognitive change that happens. We may see as we get older, there's slight decreases in our ability to respond to things. So processing speed declines. And this is normal. Everyone tends to experience this. Some people experience declines more than what's expected, but it doesn't impair their ability to function on a day-to-day basis. So it doesn't impair their daily activities, but it's still more than what's expected for normal aging. And this is called mild cognitive impairment. Now, when these declines begin to impact our daily functioning, this is when someone is diagnosed with dementia. So when these cognitive declines have become so severe that it impairs their ability to manage finances, to bathe, to cook for themselves. So if we look at this visually, you can see by the two green arrows what normal aging is. There is a slight change in functioning. So as mentioned, it might be something like processing speed, where we're not able to take in the information and respond as quickly as a younger adult. Or in memory, so our collection of past and personal experiences may start to diminish. And this is normal. Within these slight changes, though, what we see is that some cognitively older adults fall into this preclinical dementia stage. This stage is considered a silent phase, and the reason for that is because the individual has no objective cognitive change. So if they do a cognitive test, they look just as normal as any other cognitively healthy older adult. So this blue line here of someone in the preclinical stage is within the normal range. They have normal cognitive functioning despite having brain changes. So they might show neurodegeneration, they may show AD-related pathology, so Alzheimer's disease-related pathology like amyloid and tau. Despite this, still no differences in cognition. Now, in this stage, we often see that people experience something called subjective cognitive decline. So despite being normal on cognitive tests, they feel like they're struggling more. Maybe they're forgetting their keys, or they feel like they're not recalling information like they used to. And this is subjective cognitive decline. Now, as mentioned, these are normal individuals. And so if they continue to progress, they may develop mild cognitive impairment. So this is when their cognition is below what is expected for their age and their education. Again, this involves things like memory or language. So are they needing to use notes more often to remember dates and appointments and to keep track of birthdays? Are they misplacing things more? Are they having difficulties with words? So if they're in a conversation, are they forgetting the name of the street they grew up on or the name of their best friend? So we might see things like memory and language difficulties in these individuals. But again, what's important to note is that these individuals, despite the declines and despite feeling like it's a severe issue, it's not impairing their ability to perform on a day-to-day basis. So their social and occupational functioning remains intact. So again, things like bathing and dressing, eating, managing their finances all remain intact. Now, some of these individuals will stay at that level and they won't progress. However, there is a subset of that population who have mild cognitive impairment that will continue to progress. And what we see is that these individuals develop something called dementia. Now, dementia is a significant loss of our intellectual abilities that are severe enough to interfere with our activities of daily living. And as you can see on the screen here, they range from mild to severe. So in the beginning of the stages of the disease, it might be mild, where they maybe just need some help with finances or managing appointments. But because dementia is progressive, as the disease continues to get worse, what we see is that they need more and more help as the disease progresses. So we'll see, okay, in the beginning, they just need help with finances and appointments. But as they get to the severe end of the stage, they need help with everything, bathing and dressing. They need constant care. And so this progression is continuous and will continue to affect the cognitive abilities of the individual. Now, these clinical symptoms that we see, so the decline in cognition and the decline in everyday functioning, are associated with brain structure changes, right? Those structural changes that start to happen in the preclinical phase. And so these are things like increased rates of neurodegeneration, increased rates of amyloid and tau. So dementia is an umbrella term for a progressive group of disorders that are characterized by brain changes that impair our cognitive functioning. Alzheimer's disease is the most common form of dementia, affecting most of cases. And Alzheimer's disease is characterized by three main structural changes. So amyloid buildup, tau buildup, and neurodegeneration. So as you can see in the image here, we have on the top a normal aging individual compared to the bottom, which is someone with Alzheimer's disease. And just visually, you can see the large differences in the PET and the MRI scans, whereas someone who has Alzheimer's disease has much more amyloid buildup on the bottom and much more tau. Additionally, you can see here around the ventricular area, the ventricles are much larger. So there's a lot more neurodegeneration, a lot more tau, and a lot more amyloid. Recently, the Alzheimer's Working Group has decided that there are additional changes that are observed in these individuals and that they should be included in the criteria for looking at Alzheimer's disease. And this is cerebrovascular disease. So this occurs in 70% of people with Alzheimer's disease. So what we see is there's a co-pathology. They have both, the amyloid and the tau, but they're also showing evidence of this cerebrovascular disease burden. So this means that they're occurring together and that the changes in their cognition and their daily functioning are occurring as a result of both that typical AD pathology as well as these cerebrovascular lesions. Now, cerebrovascular disease is most commonly measured with white matter hyperintensities. White matter hyperintensities are more likely to occur in people with a number of risk factors like high blood pressure, high body mass index, diabetes, and smoking, for example. So these are all risk factors, both for Alzheimer's disease as well as cerebrovascular lesions. And similar to AD-specific pathology, this cerebrovascular pathology, or in the case here, white matter hyperintensities, are known to occur before the decline in cognition. So these damages and these lesions occur in the brain before there's changes in cognition. And in our previous paper, we also observed that even in normal, cognitively healthy older adults, white matter hyperintensities were occurring before changes in cognition. So if you have them, you're likely to see a decline in cognition after the fact. And so just to look at this kind of a bit more visually, I have two images up here of two 74-year-old men, one who is a cognitively healthy older adult and one who has Alzheimer's disease. And you can see the differences in the brain pretty distinctly. So if you look at the red arrows in the normal aging individual, you can see the ventricles are a decent size. They're what's expected for aging. There's some change, but not too much. Whereas if you look at the individual with Alzheimer's disease, you can see the ventricles are much larger, indicating that there's a lot more neurodegeneration in these individuals. But unfortunately, that's not the only changes we see here. If we look at the bottom picture, you can see in the normal aging individual that there may be some white matter hyperintensity. So you can see at this top green arrow, there's a little bit of kind of brightness on the screen there, and that's the white matter hyperintensities. But if you look at the individual with Alzheimer's disease, it's much more distinct, right? So in general, in Alzheimer's disease, we're seeing increased rates of neurodegeneration that is more pronounced than what we see in aging, as well as increased rates of cerebrovascular pathology, and in our case, white matter hyperintensities. Now, as I mentioned, both neurodegeneration and white matter hyperintensities occur before the cognitive changes. And that's potentially in this preclinical stage. So if we go back to this graph here, this is where it's happening in that preclinical silent phase before there's objective changes on cognitive tests. And this is that stage where people can present with subjective cognitive decline. So unfortunately, there is no cure for dementia. There's no treatment for it. And so a lot of work has gone into studying this preclinical phase, looking at if we can see the changes before they happen or when they're starting to happen so that interventions can start earlier. And so this preclinical stage has been identified as the earliest stage of dementia. And a lot of work has started to focus on that area in hopes to start interventions earlier. So as I mentioned, this preclinical stage has this subjective cognitive decline. And so that's going to be the focus of the rest of the talk, this area of subjective cognitive decline. So subjective cognitive decline is when a cognitively healthy older adult presents with normal functioning. So as mentioned, they have normal cognitive functioning, but they have this self-perceived feeling of worsening cognition. So again, they might feel like they're losing things more. They might feel like their memory wasn't as good as it used to be. So these are experiences the individual has. And unfortunately, these experiences are linked to increases in biomarker abnormalities. So those amyloid, the tau, the white matter hyperintensities, having these experiences is linked to those biomarker positivities. And because of that, we see an increased risk of dementia, increased risk of cognitive decline in these individuals. Now, there's a number of different terms used for subjective cognitive decline. So if you start looking into the research, there is a multitude of terms. There's subjective memory impairments, there's memory complaints, subjective cognitive impairment, subjective memory declines. There's a lot. And so if we start to look at the research area, it's really inconsistent and it's hard to see what the results are because there's just so many different terms. Given the number of terms, you may be unsurprised to hear that there's also a number of different methods used to assess subjective cognitive decline. So there's this one on the screen here. This is the Cognitive Change Index. It's one of the most commonly used methods, and it is a validated comprehensive questionnaire. The first 12 questions are on memory alone. So it focuses on, are you having difficulties remembering things? Are you having difficulties with new information, or potentially having people think that your memory is not as good as it used to be? The last question, so 13 through 20, focus on other things like organization and planning and the ability to focus. There are other questionnaires. So something like, do you feel your memory is becoming worse? No. Yes, but it doesn't worry me. And yes, and this worries me. So something as simple like this will be used to define subjective cognitive decline. And another study might use the validated questionnaire, and then they're comparing their results. We also have something like this, where it might be two questions. How often do you have trouble remembering things? And compared to 10 years ago, would you say your memory is the same or much worse? So what we see here is that this is just a few, right? These are just a couple of the many, many questionnaires that are used. So there's no standardized assessment. There's no consensus about the optimal measurement tools, how and when they should be implemented to measure subjective cognitive decline. In addition to that, even if two people or two researchers use the same questionnaire, they may not use the same cutoff. They might say, okay, well, if we're looking at the first 12 questions, if they get an average of 3 on all of them, we'll consider it subjective cognitive decline. Other people might use a continuous measure, so not saying yes or no, but with low to high complaints. And so there is no consensus on how we measure it. And because of this, the question now becomes, do the differences in measurement methods or in the instruments used result in differing results, right? Are there brain differences that are being observed in one questionnaire and not another? Are there pathology differences, cognitive decline differences? And there was a recent review paper that came out that had 100 at least of papers on subjective cognitive decline. And there was a number of different methodologies, right? So some studies found results, some studies didn't. And so the question is, are these inconsistencies due to the methodologies used? So for example, this is a recent paper that came out looking at hippocampal volume and white matter hyperintensity burden in normal aging, subjective cognitive decline, and people with Alzheimer's disease. And what they found is that the hippocampal volume and white matter hyperintensities were associated with executive functioning and with memory in people with subjective cognitive decline. Additionally, and importantly, they observed that those with subjective cognitive decline had smaller hippocampal volume compared to the cognitively healthy older adults without subjective cognitive decline. And that seems great, right? Okay, now we know this is the preclinical stage. However, the second paper, which came out the same year, 2 years before, found that there were no volumetric differences in the hippocampus between normal controls and people with subjective cognitive decline. Now, these two studies use somewhat similar methods to define SCD. The first one just said that they had normal cognition, but they had to report that there were differences in their memory from a previous time point, so from when they were younger. The second one used the International Working Group criteria, which again is just self-perceived deficits in cognitive functioning that differs from before. So despite them both saying, yep, there is self-perceived deficits and it is worse than what we saw previously, they had no standard questionnaire used, right? Is it one questionnaire being used or some validated technique? So they did have differing methods in how they quantified. We see similar findings when we look at pathology. So this first study here looked at amyloid burden, so beta amyloid in Alzheimer's disease. And what they found is that people with subjective cognitive decline had increased amyloid, but only if they reported that they were worried about those changes. So saying, yes, I have them, but I'm actually worried about them, was associated with the beta amyloid buildup, whereas the rest of the questionnaire wasn't associated. Now, on the other hand, this study looked at subjective cognitive decline in a continuous method. So they had a composite of 26 questions, and they found that the higher amounts of subjective cognitive decline were associated with higher amyloid. So as complaints went up, amyloid also increased. So a main issue here, just from these 4 studies I presented, is that there's a lot of inconsistent findings, and it may be because the questionnaires aren't tapping into the same construct. And so the question becomes, do these questions match up? Do they measure the same brain changes? Do they measure the same cognitive differences? And so there has been a few studies that have looked at this. So the first one looked at a subjective memory complaint scale and compared it to a memory complaint questionnaire. So the subjective memory complaint scale has these 10 questions that talk about daily life and memory tasks. So do you forget things? Are you forgetting the names of people? Do you have any concerns with your memory? And they compared it to another questionnaire, the memory complaints questionnaire, which really focused on remembering. So if you look at the 2 questions, you can really see that the memory complaints is about remembering and recalling things as opposed to potentially, are you thinking more solely? Because of this, you may not be surprised to see that there's a relatively low correlation of only 0.44 between the endorsement of questions on one compared to the other. So this lack of overlap between the questionnaires may be associated with the low correlation. A second study also looked at the everyday cognition scale versus an adapted version of the blessed memory scale. Now, the ECOG is great because it looks at a wide range of domains. We look at memory, visuospatial, executive function skills, and language. So it taps into a lot of domains. But the blessed memory scale was only 5 questions, and it focused mainly on memory. So again, what we saw was a relatively low correlation, a 0.5 correlation between 2 scales that are supposed to be measuring the same construct. So this is obviously not a good thing when we're saying that this is preclinical dementia, and someone may develop dementia if they endorse subjective cognitive decline. But what does endorsing it really mean? And so our goal in some of these papers was to start looking at differences between the questionnaires. So take groups of people who have done the same questionnaire and look at how the endorsing is happening, and is it associated with similar brain changes. Now, the two questionnaires we use have been shown to have a moderate correlation, 0.795, in the rate of endorsement in the same group of people. But this doesn't mean that the brain changes between the two are the same. So our goal was to take the cognitive change index, so the questionnaire I showed you, the first 12 were associated with memory, and compare it to the ECOG, so the one that has 39 different questions in multiple different domains. And the idea here was to examine if endorsing these questionnaires was associated with similar differences between the groups. So do brain volume differences appear in one questionnaire and not the other? Or are we seeing maybe white matter hyperintensities are associated with one of the questionnaires but not the other? And so the idea here was to look at whether or not we can see the structural differences early in the process, and if one questionnaire was tapping into it. And of course, we wanted to see whether or not the questionnaires measure the same amount, right? Are specific questions within it associated with it, or are all the questions equally contributing to the brain volume differences? So we use the ADNI data set simply just because the data is already collected, and they have two measurement tools that are used, so the cognitive change index and the ECOG. Now, we also added in worry, and the reason for this was, well, that previous study I showed you where it said worry was associated, but not the actual questionnaire. So we wanted to look at worry as well. So we had 273 older adults, and then we divided them based on subjective cognitive decline or no subjective cognitive decline based on their questionnaires. And of the sample, 91 of the individuals reported no subjective cognitive decline, regardless of the questionnaire. So they were just cognitively healthy, no subjective cognitive decline at all. Importantly, only 71 or 39% of the sample was subjective cognitive decline positive with all the definitions. So there is not as much overlap as we'd hope with different constructs. And so here we said, okay, now we see that there's a difference. There's clearly a difference in the rate of endorsement. Let's look at gray matter volume. Let's look at white matter hyperintensity differences between these groups. So before we get into the actual results, I'm going to give you a very brief overview of what grading is, because instead of looking at a volume of the hippocampus, we use something called grading. And this is really important because previous research has shown that not only volume, but things like shape and texture of the hippocampus are really important to look at when looking at memory decline and Alzheimer's disease. So we use something called SNIPE, which computes the similarity of every voxel in the hippocampus of each person to a library that has already been trained of normal controls and people with Alzheimer's disease. And so what it does is it compares not only the volume, but also the texture, the intensity, and the shape of the hippocampus, giving each person a range of, are they more similar to Alzheimer's, or are they more similar to a normal control? So just to look at this kind of visually, what we see here is in A, so on the far, let me get my laser pointer, on this far side here, we have the individuals going into the SNIPE method. So these are all our participants. What we see here is that a medial temporal lobe mask is applied to create a voxel patch around that area of interest. These patches are then fed into the SNIPE method, and it's compared to the group of normal controls here up at the top and the Alzheimer's disease. And then it comes out here at part C with an average of the entire hippocampus being either +1 or -1, in this case, red or blue, stating that the individual's hippocampus is more or less similar to a normal control or an AD. So this method is really great because it not only provides, as I said, volume, but a bit more in depth. And in our previous work, We've also shown that this method is better at detecting, or not detecting, but classifying participants into Alzheimer's disease, into normal controls, or people with MCI. We also saw that this method is sensitive to episodic memory changes in individuals with subjective cognitive decline. So we focused on this grading method instead of just volume alone. And so that is what hippocampal grading is. And now that you have a general idea, I'm going to start to tell you the results and how they differed between the questionnaires. You're welcome. Okay, so for the cognitive change index, so people who endorse subjective cognitive decline showed smaller left hippocampal grading. Now, this means that the other regions were not different. But when we look at the ECOG, we see a lot more widespread, right? And this may just be because, as you may recall, the ECOG looks at memory, language, executive functions, visuospatial abilities. And so we see differences in the right hippocampal grading, right amygdala, and both the left and right superior temporal regions. When we look at ECOG and worry, we see lower left hippocampal grading. And then when worry alone, we see lower left hippocampal grading. So as you can see here, rates of volume differences, this pattern between the groups varies depending on questionnaire. Now, we also did something similar looking at white matter hyperintensities. And this is a great technique developed by your very own Masa Dadar here that looks at white matter hyperintensities in T1 weighted images. So normally you have to use T2 or FLAIR images, but this technique works really well in T1 weighted. And so we were able to use all of ADNI, which is a great benefit because not everyone in ADNI has a T2 or a FLAIR image. So props to you, Masa, for developing such a great technique and allowing me to do this research. So when we look at the cognitive change index, what we see is that the temporal and parietal regions differed in white matter hyperintensity burden. So those with subjective cognitive decline had increased white matter hyperintensity burden in temporal parietal regions. When we look at the ECOG, very similar, temporal and parietal regions again. When we look at the ECOG plus worry, we see total, frontal, temporal, and parietal regions differing between the groups. And then with worry again, total, frontal, temporal, and parietal. So if we look at the image here, you can see that this is for total white matter hyperintensity burden. And there's a lot of overlap in both the CCI and the ECOG. And we start to see the differences between the groups when we look at worry and then ECOG and worry. So this is really showing again, now not only neurodegeneration differs between, but also white matter hyperintensities. So with these results, in summary, we're seeing that if we look at the cognitive change index, you're going to see neurodegeneration in the left hippocampus. We're going to see increased white matter hyperintensity burden in the temporal parietal regions. Whereas if we do ECOG, we're going to see more widespread neurodegeneration, superior temporal regions, hippocampus, amygdala, as well as those temporal and parietal white matter hyperintensities. And then, of course, when we get into ECOG and worry, we're looking again at the hippocampus for neurodegeneration, but a more widespread burden of white matter hyperintensities. And similarly with worry, again, hippocampus and a widespread burden. And so from here, we further thought, well, is it specific questions? Are there certain questions on these tests that are driving these differences? So if someone endorses question 1, is that the one that's associated with all the neurodegeneration? Is question 2 associated with all the white matter hyperintensities? And so the next part was to further examine the questionnaires themselves. Now, ADNI has continuously updated. So now we have 332 cognitively healthy older adults who've completed both questionnaires. And we compared the questions from both questionnaires that assess only memory. So we're looking at the 12 questions from the Cognitive Change Index and the 8 questions from the ECOG that assess memory. We then used partial least squares to compare the questions in the data. So the idea here is to look at if there is an underlying or hidden variable underneath the data that can be inferred to allow us to see what is driving the differences in the questionnaires. And so by exploring these latent variables, we can see which questions are most related to these changes, which can help us obviously with predicting and understanding changes that are occurring in the brain. So from these results, right, we took those 20 questions, the 8 ECOG, the 12 CCI, and we looked at volume differences in white matter hyperintensity burden. And in doing so, what we see is that the changes are mainly associated with the cognitive change index, and specifically a couple of questions. So question 2, 6, 7, 10, and 12. So despite there being a number of questions, the 86% of the variance observed is due to these 5 questions. So these 5 questions are associated with memory for faces, new information, and memory for notes. So it's really focused on that memory aspect, whereas the questions that aren't involved are more associated with recalling information and remembering what someone is intended to do. So there is some potential slight differences, whereas the question involves are very concrete focus on memory, and the questions that aren't may be tapping into other aspects than just memory. So there are some other factors that contribute to subjective cognitive decline and changes in age, one being biological sex and the other being education. So females have more to cognitive decline, unfortunately, associated with subjective cognitive decline. So our previous work has shown that when males and females both endorse subjective cognitive decline, females have more cognitive decline over time. So they decline at a steeper rate if they endorse it compared to the male counterparts. They also exhibit more Alzheimer's disease related pathology. So that amyloid and tau burden, putting them at an increased risk for Alzheimer's disease and dementia. Similarly, with education, there has been a lot of work that has looked at the effects of education in subjective cognitive decline, with more highly educated individuals being more sensitive to their changes. So if someone who is highly educated has reported subjective cognitive decline, there is a stronger relationship with cognitive change, as well as with like amyloid and tau buildup. So both education and female sex may play a role. And so we further looked into this PLS analysis, subdividing based on male and female sex. And again, we see something that coincides with our previous work, that the questions in females are more associated with reduced volume and increased white matter hyperintensities, whereas in males, that relationship. So again, we saw something very similar with education. The same questions are going to come in at the item level or at the brain level. So we will further look into this. But it's really interesting to see kind of the take home is that the 2 questionnaires may not be associated with the same types of changes. So we see that the 2 questionnaires are not associated with the same amount of neurodegeneration and white matter hyperintensities, right? When we look at the PLS, and when we look at the first LV, we see that mainly the cognitive change index is associated with those changes. And then, of course, we see differences in high education versus low education in the males versus females. So potentially these questionnaires are not doing what they're supposed to in different groups of people, which has really important implications for clinical and research settings, right? So if we look at a clinical setting, if we're implementing a questionnaire that potentially doesn't measure what we want it to, it's potential that maybe some of these questionnaires look at frontotemporal dementia, or they're tapping into vascular, where the other one may be looking at Alzheimer's disease. And so we shouldn't be using these questions interchangeably, and more research is definitely needed to uncover what is going on with these questionnaires and why are they not tapping the same thing. And so our future research is going to continue to look at subjective cognitive decline, postmortem differences, brain changes across different ethnicities and cultures. Do the questionnaires tap into the same thing across cultures? And then, of course, different research environments. So if someone is part of a cohort study like ADNI versus a memory clinic sample, is one more associated with change than the other? So there's still a lot of research to be done on this, especially because it's been identified as that preclinical dementia phase. So we really need to find out what is most important, what changes are happening here, and why are they happening before we can start with any intervention. So I just want to send out a few acknowledgements and thank you to everyone who's participated and supported me as we do this. And thanks so much for having me here.